Well, greetings, students. It's been a whirlwind, hasn't it? So in these last few screencasts, we sort of started with this basic overall reaction for cellular respiration. Glucose plus oxygen yields CO2 and water. And we've seen it's actually much more complicated than just this single overview reaction, right? But hopefully by now you have a better sense of the steps taking place through glycolysis, pyruvate oxidation, and the Krebs cycle as this glucose is broken down into CO2. And we've seen that the CO2 is generated during pyruvate oxidation and the Krebs cycle, and it's exhaled as we breathe. We've seen that the oxygen comes in during the electron transport chain as that final electron acceptor. And we've seen that that water is produced when the oxygen accepts the electron. The oxygen accepts the electron to become water. And so we basically accounted for this entire overall reaction, and we now understand in more detail than we ever wanted to know how that ATP is produced. And again, you may need to rewatch some of these screencasts and review it because it is complicated. But again, focus on these large principles, exergonic reactions being coupled to endergonic reactions, et cetera, et cetera. So to wrap up, a couple special cases. We've been sort of focused on the mitochondria, which means we've been focused exclusively on eukaryotic cells. What about prokaryotic cells who don't have mitochondria? Can they do cellular respiration? And the answer is yes. Yes, they can. So here's a diagram of a bacterial cell. So if we zoom in on this bacterial cell, basically what we see is the same electron transport proteins that we saw in the inner membrane of the mitochondria. It turns out that in the bacterial cell, though, it's just that instead of using the inner membrane of the mitochondria, they just use their cell membrane as that membrane. And that should make sense because the endosymbiotic theory says that a bacterial cell was taken in by a larger cell. And so the cell membrane of that bacteria ultimately would become the inner membrane of the mitochondria. And so glycolysis in a prokaryotic cell still takes place in the cytoplasm. The citric acid cycle, it turns out, also takes place in the cytoplasm of a prokaryotic cell. Because that cytoplasm of the prokaryotic cell ultimately became the mitochondrial matrix. And the electron transport chain just takes place in the cell membrane of a bacterial cell. And so the proton gradient is actually formed across the inside and outside of the cell itself. So bacteria do this same process, just a little bit different. So here's the answers, and make sure you are familiar with how this works in prokaryotic cells. The other thing I want to mention is, what about other foods, right? So we've been focused exclusively on glucose, but I think you know that you also can use fats for energy. You can even use proteins for energy. So if you eat a big old, what is that? Is that a Whopper? I don't know. It looks good, whatever it is. There's going to be fats and proteins in there as well. So here's a diagram that kind of shows how other molecules feed in. So carbohydrates, when you eat like the bun of that burger, ultimately that bun is broken down into sugars, which then can become glucose and feed into glycolysis directly. What about these other things, fats and proteins? Well, fats, you may remember, triglycerides are basically this 3-carbon glycerol with 3 molecules of fatty acids. And then proteins are made of amino acids. And we can see that these things feed in to various steps of this. So how does that work? Well, let's start with fats. Fats undergo something called beta oxidation, where basically those fats, these long hydrocarbon chains, are cleaved into 2 carbon units. And you can see that here, 2 carbon unit, 2 carbon unit, 2 carbon unit, 2 carbon unit. So we just, through this beta oxidation process, we just chop this fat into 2 carbon units. And then those 2 carbon units feed into the Krebs cycle as acetyl coenzyme A. And so a single fat molecule could give us 8 different acetyl-CoAs, which is going to give us a lot of NADH and FADH2, which will produce a lot of energy. And I think you know that fats are used to store energy and that they give us a lot. And this is why there's a lot of these 2 carbon units that can be used. Proteins, a little bit more complicated. proteins are broken down into amino acids, and each amino acid feeds into a different place. But the one key idea that is true of all amino acids is something called deamination has to occur, meaning those amino groups are broken off the amino acid. So each amino acid has that amino group, and the amino group has no role in energy production. So it's just broken off and it's removed, and ultimately you urinate it out. So generally your body doesn't tend to use proteins for energy. It tries to use sugars and fats, but if it does start to break down proteins, it's going to break those off. And then this carbon skeleton, and the carbon skeleton is going to vary in length depending on what that R group is. That carbon skeleton will then feed into glycolysis or the citric acid cycle at some point. And again, it's different for every single amino acid, so that gets quite complicated. So yeah, the gist is these are just broken down into intermediates in these processes and can also be used for energy. Okay, so there's just another summary showing the triglycerides feeding in there. The final topic I want to address is something called fermentation. So it turns out that cellular respiration is only one mechanism by which we produce energy, and it's limited. So one problem with cellular respiration is it's slow. You've got 10 chemical reactions for glycolysis. You've got another reaction for pyruvate oxidation. You've got 8 more for the Krebs cycle. You got all this electron transport chain business. That takes a long, it's slow. So what happens if you're doing like the 50-yard dash? You know, you hit those starting blocks and you sprint and you're just tearing out of there. And after, 12 seconds, you're done. Well, there probably wasn't enough time to really get cellular respiration going. You need something else to meet that increased demand for energy during that sprint. Fermentation is that something else. Another limitation of cellular respiration is it is aerobic, meaning you need oxygen for cellular respiration to take place. I think we've all experienced being, on a run or maybe walking up the stairs at the school where you're sort of like, all of a sudden you aren't getting enough oxygen and you're kind of like, you're gasping for air. When you gasp for air, that means that your body is struggling to get oxygen to your muscles to keep up with demand. So whenever you're gasping for air, you are no longer aerobic, you are in fact anaerobic, meaning your muscles aren't getting enough oxygen to keep them going. To be clear, you're not oxygen starved overall. Your brain has enough oxygen, you have enough oxygen to live, but your muscles don't quite have enough to keep up with that. And so this is called hitting the wall in a marathon or something, where all of a sudden you're just not getting the oxygen you need to keep this up. So if cells don't get enough oxygen, again, what happens is this oxygen can no longer accept that electron. And that is problematic. And I'll tell you why at a biochemical level. If the oxygen can't accept the electron, this protein can't get rid of it. So this protein becomes jammed up. It's got an electron, but it can't give it to oxygen because there isn't any oxygen in their muscle cells. Well, that means that this protein gets jammed up because it can't pass its electron there. And then this protein gets jammed up, and then this protein gets jammed up. So we basically have this backlog of electrons that can't be passed, and ultimately NADH can't pass an electron on, and this gets jammed up. And that is problematic because if NADH can't give an electron off and reform NAD+, well then, over here, NAD+ is not available for glycolysis to occur because this reaction requires that NAD+ to form NADH. So if we don't have oxygen, this whole system backs up and we're going to have a whole bunch of NADH, no NAD+, and without NAD+, glycolysis stops, the Krebs cycle stops, everything stops, and we're in a huge mess of trouble. And so this is where fermentation comes in. Fermentation is a process that regenerates this NAD+, so that at least glycolysis can continue. And you can see this over here. So this NADH that's formed in glycolysis can actually be converted back to NAD+ by these steps here in fermentation. And if NAD+ is reformed here, then it can be used in this reaction, and this overall process can continue. So fermentation is basically something your body does to just keep a little bit of energy going. If there's not enough oxygen for cellular respiration, then by golly, at least we can keep going through fermentation. So here's just kind of a zoomed in picture showing glycolysis and that relationship, how NAD plus is used here and NADH is generated, and then NADH is used in fermentation to regenerate NAD plus so that this reaction can continue and glycolysis can continue. So basically, after glycolysis, the fate of pyruvate really depends on the environment that the cell is in. If it's aerobic, that pyruvate is going to move into the mitochondria, the citric acid cycle occurs, the electron transport chain occurs, and everything we've just talked about is going to happen. If, however, the cell is in anaerobic conditions, then the pyruvate stays in the cytoplasm and fermentation occurs. There's basically two ways fermentation can happen, and it depends on what species you belong to. If you happen to be an animal, such as a human being, you do lactic acid fermentation. Whoops. And in lactic acid fermentation, this pyruvate is converted to a molecule called lactate, and that lactate is a waste product that is later broken down by the body, and the purpose of it is to take this NADH, regenerate NAD+, so the glycolysis can continue. If you happen to be a yeast or certain bacteria, you do alcoholic fermentation, where you take that same pyruvate, but now you're going to convert it to ethanol. Now, ethanol is only 2 carbons long, which means a molecule of CO2 is lost along the way. But again, key step, the NADH is converted to NAD+, so glycolysis can continue. So the key to both of these is this regeneration of NAD+, and then it's just the waste product that differs. Here's why this is cool. Lactic fermentation, lactic acid fermentation, you may have been told that this is what causes muscle soreness. Your coaches might say, oh, you got to flush the lactic acid from your system. Turns out that's not true. The soreness is actually a result of little tears that happen in your muscles, and then your immune system kind of kicks in, the area swells, and the swelling pushes against your nerves and causes that soreness. But you may have heard your coaches talking about lactic acid, and they are right that if you're doing, sprints or short, intense bursts of energy, you tend to use fermentation for that, and then that causes those muscle tears and soreness. Alcoholic fermentation is useful for lots of things. So bread making, you put yeast in bread. Yeast are little tiny microscopic single cell organisms, and they're related to fungi, and they do lactic acid fermentation. And you may have heard that yeast causes bread to rise. And the reason why the bread rises is because the pyruvate is broken down to ethanol and CO2. And so little yeast that get kind of trapped in the dough produce CO2, that gas is produced, and that makes the bread rise and expand, and then you bake it, and you harden it, and you get this nice, airy, fluffy bread. Alcohol fermentation also produces alcohol. So the ethanol piece is also produced from fermentation. You may have heard of, we're going to ferment the grapes to make wine. And so that ethanol is then harvested and purified and used to make alcoholic beverages. And in fact, the different kinds of alcoholic beverages are formed by giving the yeast a different starting material. And so here's a little, you can see, how much do you know about alcohol, underage high school minors? So you can do a little quiz here, and I'm actually not good at this. So wine is grapes, tequila, what is tequila? I think that's potatoes. Beer is barley. I might be getting these wrong. I don't really drink much at all, really. Tequila is agave, and vodka is potatoes. What's rice? Sake, that's rice. Okay, well, there you go. Anyway, so that's it. So basically, to recap, if you're on a run, if you're doing a lengthy exercise, and you're breathing comfortably, you're doing cellular respiration. You have plenty of oxygen. Cellular respiration is powering you. But once you hit a hill, or you do a sprint or something, and you start to gasp for air, that means your muscles aren't getting quite enough oxygen, and so you shift to fermentation. And since fermentation only produces a little bit of glucose, you're probably going to get tired more quickly once you're gasping for air. So you slow down, catch your breath, boom, cellular respiration kicks in again, and you're off and running. So that's kind of how it works in the real world. Also, if you're a sprinter, there's not really time